So I'm from the Danish Cancer Society in Copenhagen, Denmark, and we are a non-profit, non-governmental organization, and we are funding most of the cancer research in Denmark. Just to remind you, where Denmark is a small country in the north here, if you don't, and we are actually not that small because Greenland also belongs to in principle to us, and you can wonder why they call it Greenland now. It's so wide, but I'll not go into this. This talk will be about plasma membrane repair in cancer. This is a new concept that we have recently found may play a significant role in metastasis. So as you know, it's quite essential if you dive in a submarine or if you are a cell to maintain integrity of the of your surface, and this is very important for cells to maintain plasma membrane integrity. Traditional plasma membrane repair has been considered as a, or originally it was assumed that plasma membrane was facilitated by fusion of vesicles to the wound, and the trigger of this fusion was calcium influx into cells, and then you would have a fusion of different vesicles around the injury site, mainly lysosomes, that would eventually heal the wound. However, this model is a bit simple, as we now know that there are many components in the plasma membrane repair system, and many of these components seem to be upregulated in cancer. Navigating through a dense extracellular matrix is challenging for cells and also for cancer cells. If you look here, cells will migrate out in two different types of collagen. However, if you add this broad inhibitor of metalloproteins inhibitor GM6001, cells are still able to migrate out, and if you look careful, they actually, when you add this drug, they come more into pieces, but they still seem to be able to walk out even though they're not able to use their secretory enzymes anymore. We also observed when you look carefully on cells that navigate through collagen that that sometimes they lose a part of their membrane, but they still manage to walk through the matrix, even though they lose part of their cell body. So somehow they must have an efficient plasma membrane repair system to cope with these injuries. We have used a cell system based on the MCF7 cells that can be triggered to overexpress a truncated form of HER2 or ERBB2. This cell system is reflecting quite well what you see in expression arrays and proteomics in these HER2 overexpressing cells. And these are very dynamic cells. They will move around, even crawl on top of each other, and they have a lot of dynamic in the plasma membrane. However, the surprising thing was that if we looked at their ability to maintain integrity of the membrane just under steady state conditions, then these cells that have this enhanced motility, they take much less up of impermeable dye compared to the control cells. So this suggests that they might have an enhanced repair system. And of course, here we have control for endocytose activity as well. Annexin A1 has previously been described to be a protein involved in plasma membrane repair. So we use this protein to assess if this enhanced membrane dynamics would recruit Annexin A1 to these protrusions. And you can see sometimes Annexin A1 is actually accumulating out in the edges, and this is most likely a sign of injury to the membrane that they cope with by recruiting this protein. Now, in order to find more of, regulators of membrane repair, we did a proteomics where we harvested plasma membrane fragments from cells after injury, either detergent-induced injury or scrape-induced injury. Then we analyzed by quantitative mass spectrometry. And there we found a small protein, calcium-binding protein, S111, which is found hugely overexpressed in a lot of different metastatic tumors. It's also overexpressed in our model system here. And when we remove S111, these cells are not able to navigate through matrigel anymore. So it seems to be important for the ability to move out. In some cell types, it even induces cell death if you remove S111. We went further with this protein, and we looked at the integrity of these cells overexpressing S111, the invasive ERB2 overexpressing cells versus control cells. And if we injure the cells by a focused laser here and here, you can see that these cells are not very good at repairing compared to these ones that seem to repair very fast and excise a part of the membrane as a part of the repair system. We could see that when we remove S111, then cells were much poorer at repairing plasma membrane injuries compared to control cells. We have done a lot of different torture to cells, rolling glass beads upon them, and all these assays show that they cope much better if S111 is present. Now, these laser injury experiments were used to address the recruitment of different repair proteins after injury. And again, we used laser injury here. You can see that these cells overexpress Annexin A1 and then S111. Annexin A1 has been described as a binding part of S111. However, when we do laser injury here, Annexin A1 green is going directly to the site of injury, whereas S111 is actually defining the repair zone where the cell chooses to cut off the injured part of the membrane. You can see a similar example here. S111 in red is defining the repair zone and where the cell chooses to cut off the damaged part of the membrane. If you're really harsh to cells, you can even damage them in both ends. And we do that. We did that here, and you can see that they eventually can cut off both ends of the cell, and the cell is, although struggling, but it's still able to recover from such an injury. So they have a dramatic ability for wound healing. If there's no calcium present, then the cells will not be able to repair at all. So these repair systems are dependent on calcium. This was just a quantification of our movies. Now, we're of course looking at mechanistic. How does this system work? And we used we combined then S111 with different annexins, and we found actually that annexin A2 was most likely the binding partner of S111, as they seem to co-localize around the site or the repair site. We can also see that by immunoprecipitation that we could pull down both proteins together. As mentioned, the S111 protein, they bind and cooperate with annexin proteins. Annexin A1 here is an example where you see that calcium binding will form a concave site that allows the recruitment of this site to the membrane, and then this structure can facilitate annex or glue membranes together. And we found that annexin A2 was required for bringing S111 to the plasma membrane. If we remove Annexin A2, then there's no S111 at the plasma membrane, and vice versa. We also did some mutagenesis gymnastic where we mutated the N or the C terminus. So the C terminus does not have a significant impact on the translocation of S111 to the membrane, whereas the N terminus has a dramatic impact. We see a localization of S111 of Annexin A2 on the surface of the plasma membrane. Now, actin has been shown to be involved in wound healing in many, not many, but in some cell models, xenopus oocytes, where there's formed a actin, myosin actin constriction ring around the wound. So we thought that maybe Annexin A2 S111 is regulating actin modulation around the wound parameter. And when we roll glass beads around on the top of cells, we see that both beta-actin and S111 is co-localizing around the wound. And the fact that actin is important for wound healing, we can see here, this is a quite harsh injury. We see that the cell is struggling with a repair, but eventually it builds up a huge actin zone here. A cell is still able to survive this injury. When we overexpress beta-actin together with S111, you see that S111 in red is going to the site of injury and co-localizing strongly with the fibers of beta-actin here around the wound parameter. We also, of course, we depleted S111, and then we looked at actin recruitment to the site of injury. So in control cells, you will see the recruitment of actin around the injury site, whereas without S111, there's no recruitment. And if we mutate the calcium binding domain of S111, then there's no repair or there's no response to this injury. And the cell here will eventually die. Now, I'll just here and then show you. So this is how in a healer cell with a protrusion here, where we injure it in the end, you'll see first waves of S111 building up, and then followed up by actin polymerization, and then eventually healing of the cells. And this is something that cancer cells, and especially these HER2 overexpressing cells, are very good at. And we believe that the plasma membrane repair system might be an attractive new target for hitting cancer cells. At least it has not been explored before. This is our current model. We have, upon an injury, calcium flushes into the cell, and S111 and Annexin A2 is recruited to the site of injury. This complex facilitates somehow the modulation of actin buildup around this injury site, and allows the membrane to be pulled together. I should also mention that one of the first events that happen when you have an injury is actually that the membrane is relaxing, and this relaxation is necessary to actually, because you have a cytoskeleton beneath the membrane, so the first step is actually relaxation of the membrane, where you then can bring the edges together. And the Annexin A2 S111 is probably building up actin after this relaxation has occurred. So this model is not completely accurate because, as we also saw, we also see injury in the end of when cells are navigating through extracellular matrix. We also see that in the end of the cells, you have injuries and the cells are shredding off pieces of the membrane. But to cope with the harsh tumor environment or the extracellular matrix, you will need to constantly repair proteins, repair a membrane, and this is facilitated by the Annexin A2 S111 complex. As we know, science is teamwork, and many people have contributed to this work. I would especially mention Jody Eiswell from Children's National Medical Center in Washington. He's a magician with a confocal microscope. And we are funded by the Danish National Research Foundations from Health Sciences and Natural Sciences, and we also get a bit of money from the Danish Cancer Society. Thank you very much for your attention.